Hi everyone, and welcome to the 19th TIA Centre seminar of the academic year. And I'm absolutely delighted to introduce today Professor Asa Ben-Hur, who of course is Pfizer's or was Pfizer's supervisor and will be grand supervisor to many people around here. So Asa is Professor of Computer Science at Colorado State University. His research is at the intersection of machine learning and computational biology, with a current focus on deep learning applications in genomics and the interpretability of these models. His work has been published in leading venues in computational biology, and machine learning. His research funding has totaled over $4 million, and he was co-PI on a $3 million NSF Research Traineeship award to develop an interdisciplinary graduate program at the interface of biology, engineering, and computational sciences, and more recently, co-PI on an NIH T32 training grant. And the title of the talk today is Foundation Models in Genomics, a supervised alternative to DNA language models. Asa, thank you very much for joining us. And when you're ready.
Technical difficulties. Anyhow, this is just a piece of genomic sequence, and yeah, for us as computer scientists, well, this is just a string. And what do we do with strings? Well, we train models, and well, you might ask, is there a ChatGPT for the genome? Well, the answer is yes, and actually quite a number of them. But these DNA large language models, well, they basically are trained on lots and lots of DNA sequences. So same architecture, same way of training a language model, exactly the same way you train a human language model. Except, well, the downside is that you can't interact with it the way you interact with ChatGPT. And so there's been a lot of interest in the community. Well, what can we do with these DNA language models? But what I'm going to share with you today is a different approach that uses supervised learning rather than unsupervised learning. And the point I'd like to make is that, well, human language, the reason why, let's see, why large language models work so well is that human language is kind of self-contained, that apparently all the information that's needed is there in the text. But with DNA, it's very different, because it's not just a sequence of A, C's, G's, and T's. And yeah, you've had all some exposure to biology, and you know it's complicated. The DNA, in fact, has a state. There's epigenetics. There's all the multitude of molecules in the cell, and it's that combo that gives DNA its meaning. So the sequence itself, well, there's a lot that can be learned from it, but at least my intuition is that to make progress on modeling biological phenomena, we're probably not going to get there simply with DNA language. And just back to our DNA sequence, and well, what can we, yeah, it just looks like a undecipherable combination of nucleotides. But yeah,
you finish your sentence in the next question.
So there's actually a lot of signals within the DNA that guide its function. So what you'll see here is some there's a promoter region, there's exons, introns, so you can see where's my GTAG? PG, yeah, so KG here, GT here, that kind of mark and an intron. And there's upstream signal, downstream signals, places where transcription factors might bind, and so on and so forth. So there's definitely information there that is there for us to detect.
I was just gonna ask, for normal LLMs,
Mhm.
you use pretext tasks, it says generally fill in the blank,
Yes,
and you know what that blank is.
yes,
So in this case, and as the model learns, it gets a fairly good idea of what that blank might be,
yeah,
right? Because it learns about the structures, learns about the grammar, learns about, all the intricacies of the human languages.
right.
With this, what are the pretext tasks normally?
So it's still fill in the blank,
Okay,
and the blanks here are, well, the tokens in regular LLMs. Yeah, they're just words or parts of words. So here, the tokens are k-mers. Typically, yeah, the simplest thing is to take, let's say, k-mers of length 4 or 5. That's been the basic approach, to take fixed length k-mers. There's more sophisticated ways of doing it with variable length tokens. So tokens, they call that bypair encoding. Yeah, the tokens that are typically from 1 or 2 to 5 nucleotide long. It's just a way of kind of optimizing the space of tokens. But it's the same exact objective, fill in the blank.
okay.
And there's, so the information it can use is, well, when it's a coding sequence, then there's a lot of this functional constraints on that. In non-coding regions, well, there may be fewer constraints, but typically these models are trained on many genomes, not just the first, the DNA bird, I think it was trained just on the human genome. But nowadays, typically the approach is to train on as many genomes as you can find, which is a lot.
Mhm.
And so seeing the same context in a lot of related genomes, that's what enables these models to learn. Plus, yeah, these, like regular LLMs, some of these are really large, billions of parameters. So the same kinds of challenges in terms of using and fine-tuning regular language models are present here. So we're going to present a lightweight model that is trained in a supervised way that basically gives us better performance at downstream tasks. So yeah, just a wee bit of, there will be biology sprinkled here and there. Yeah, how many of you know what a transcription factor is? So, not very many, so this is an important concept. So I'll go through it. And so how does a gene know that it needs to turn on or off? So upstream of the gene, there's typically something called the promoter region. And basically what happens, think of it as like a active a area of the gene that has on and off buttons that by binding in those upstream regions in their binding sites, transcription factors can either activate transcription of a gene or repress it, and often cases you need multiple of these transcription factors to bind in order to activate transcription of a gene. So think about it as a kind of the logic that guides transcription of genes. Now, what one of the projects I'm going to describe is actually recent work that we did that shows that these DNA binding proteins also have a function in RNA splicing, but we'll get to that later. So everyone in this room knows convolutional networks. So the first thing I'm going to describe is, and this is convolutional networks are still one of the primary tools used in deep learning in genomics. Well, images are two-dimensional objects. A DNA sequence is a one-dimensional object. So instead of doing 2D convolution, you do a 1D convolution. So we have, so what these convolutional filters, they're typically, in 2D, if it's just a single channel, then it's a 2D matrix that you just slide around the image. Here, our input to the model is going to be a representation of the sequence. So every nucleus, so we have 4 nucleotides, so you can represent a nucleotide by a 4-dimensional vector that represents the nucleotide in that position. And same as in computer vision, you want to detect local features. So those local features might be binding sites for transcription factors, for example. So transcription factors bind a short sequence in the DNA that's typically something like 5 to 15 nucleotides long. And so that tells us that, okay, we'll just use a convolutional filters that are around that size and slide them along the genome, and that's our 1D convolutional operation. And of course, like in computer vision, you can stack multiple of those to have a deep network, although the first application of this, they called it deep learning, but it was just a single convolutional layer.
Sounds fancy. Am I right in thinking that by representing your sequence in that way,
Yes,
as you have illustrated,
yeah,
that every nucleotide is a 4-dimensional vector in itself?
right.
that by representing it that way, you're representing it as a long image with very small height.
Yes.
Okay.
So just kind of have see how it might work. So here's our input sequence converted to a one hot encoding. You have your 4 by something convolutional filter that represents some kind of genomic signal, and then you apply this convolution operator along your sequence, apply your favorite activation function, and yeah, there's even max pooling, and then on to the next layer. So that's our basic building block. And as I mentioned, yeah, so 2012, you know what happened in 2012. So folks in bioinformatics took notice, and 2015 we had our first application of deep learning into train these convolutional network to predict binding profiles of transcription factors in DNA, and they showed that this worked a lot better than any other approach before that.
So before that would be all string-based kind of approaches.
Yeah, and well, the simplest thing that you can actually do is represent this, let's say, basically represent the binding profile by what's called a weight matrix that is basically, each of these is let's say a vector of length 4 that at each position it tells you what are the binding propensities for each of the 4 nucleotides. So that's kind of the most naive way of doing that. Although there are correlations between positions, and it's there are more sophisticated models, but this seemed to work a lot better than anything people have tried until that point. Now, okay, we need another bit of biology. Yeah, you've all might have heard about epigenetics, but just give you a little bit of an overview of what that means. So the human genome, if you stretch the DNA sequence, it'll be almost 2 meters long. And you need to fit all of that in the nucleus, which is just maybe a few microns. And you need to stuff all of that in there and still maintain the functionality of the genome. So what happens is that the DNA is wrapped around these proteins called the histones. And it's all tightly packaged nicely into the nucleus, and that's this combination. So this collection of these histones with DNA wrapped around them, that's called a nucleosome. And this overall combination of proteins and DNA with wrapped around with histones and nucleosome, that's called chromatin. And there's a little more to it in that the histone proteins can have all sorts of biochemical modifications. There can be methylation events of the DNA, and all of these changes. Yeah, we talked about transcription factors as on-off switches that affect how genes work. Well, this is another layer of that. These are all like little switches that, again, contribute to the logic that controls how the genes work. And so one of the, you guys probably all know that, yeah, sequencing is incredibly cheap and you can do amazing things with sequencing, including probing all these different aspects of chromatin. So you can determine in a high throughput way where are all these transcription factors binding. You can also get information about positions where the DNA and the histones are modified. Now, when the DNA is wrapped around histones and nucleosomes, it's actually, a gene can't be transcribed. The chromatin needs to be unwrapped a little bit in order for transcription factors to bind and for a gene to be transcribed. So knowing what parts of the genomes are accessible is incredibly useful because it tells us, okay, this, in order for a gene to be activated, the promoter region is typically needs to be accessible. So understanding gene regulation is, in order to do that, we need to have a good idea of how chromatin works. And so all these high throughput assays, well, for example, if DNA accessibility, do I have a, yeah, so here's an example of how that data might look like. So DNA-seq, that's the fancy name for this sequencing assay that probes DNA accessibility. And so the sequencing reads that you map to the genome, how much coverage is an indication of how accessible that genomic region is under certain conditions. And then this ChIP-seq stuff, that's basically transcription factor binding, and you can see that this occurs in the vicinity of this accessible region. And in the ENCODE project, yeah, they've assembled mountains of data for all these different modalities. And so again, this is going back to 2015, 2016. People designed these deep learning models in order to predict chromatin state. These two in particular were for predicting open chromatin. So regions where we have these peaks in the coverage of this DNA-seq experiment. So you'll have, so this Bassett model, for example, they use data from 170 something different experiments. And so you'll have, it was a 3-layered convolutional network that you're trying to predict 170 something outputs. So sequences of length around 600, trying to predict what's going on in terms of DNA accessibility in those regions. Yeah. Any questions so far? All right, so fast forward a few years and we have this model called SAY, and this comes from the same group that came up with Deep Sea. And rather than just predicting chromatin accessibility, they predicted over 20,000 different outputs. So from 170 something to over 20,000. So here they're predicting transcription factor binding, histone modifications, so whatever they could lay their hands on, and basically going from sequences of a length 4,000 something to what's happening to these 20,000 outputs that represent what's happening in that 4,000 nucleotide region. So that's all well and good, what I'm going to show you is how we've leveraged this same model for some for a problem that we've been interested in. Yes, ma'am.
So 4,000 nucleotides and
Yeah.
predicting 20,000 variables.
Yes.
Is that enough information in that 4,000?
Yes and no.
4,000 four-dimensional vectors.
I think it gets worse. Nowadays, there's models that will do that for sequences of length 200,000. So, of course, yeah, this same model is a purely convolutional network. Yeah, these models that work on windows of length 200,000, yeah, that's transformers. But to answer your question more specifically, well, you do that at every location in the genome. So, but yeah, you are going from, yeah, the output space is pretty high dimensional. But think about it as basically a large multitask learning problem. So you have 20,000 related tasks that leverage the same representation. So you're learning a joint representation for predicting these 20,000 different variables at each position.
Where do you think you hit the limits of predictability? That in 400 nucleotides, so basically from entropy point of view, 4,000. So entropy point of view, all you need is 2 bits represent each of them, right? So you got 4,000 2-bit information carrying data points. So 8,000 bits of information.
Well, that's a lot of bits.
Yeah.
So are you asking, is there enough to constrain this? Well, that's the proof is in the pudding, as they say. That it works, and it works quite well. And I'll share with just what sorts of things can we do with these models? Because yeah, this is biology. Just predicted, just being successful in predicting all this stuff is nice, but it's not why we do this. We want to, what can we learn about biology from these big black boxes? And so part of our research is to come up with ways to learn from these models. And now the simplest thing that people have done to try and figure out what these models are doing is to basically interpret the convolutional filters as patterns that the transcription factors find at. So that's the simplest form of interpretation that people have applied to these models. Another interesting thing you can do with these models is, well, you have a long genomic sequence, it's doing something. Well, what are the key positions in the sequence that are important to its function? And that's where you can use some of your favorite gradient-based methods to find important positions, and presumably mutations in those positions could be critical. Now, when you have, let's say, a bunch of transcription factors binding in a genomic regions, they typically work in tandem with each other. So what we did then was to look at self-attention layers and interpreted the self-attention matrix as providing us information about interactions between transcription factors. So we basically applied a statistical test essentially to the results of the application of attention, and got some interesting results using that. And in a follow-up paper, my student also added a entropy term to an entropy penalty that sparsified the attention matrix to make the attention matrix even more interpretable, so that it really stands out what the relationships that the model is picking up on. So at this point, we'll jump into the first project that I wanted to describe, work with a PhD student in the lab, describing how we can use these supervised chromatin state model as a platform for developing a method for understanding related gene regulatory phenomena. In this case, something related to what's called intron retention, which is a form of alternative splicing. So again, back to a little bit of biology. So how many of you have heard the terms introns and exons? So not everyone. So when you look at a gene, so you know that the mRNA gets translated into protein, but, and there is a big but, in eukaryotes, there's something that happens before that, and that is splicing. So your DNA gets transcribed into pre-messenger RNA, and these blue boxes, they're called exons. These salmon colored boxes are called introns. And what happens is that the introns get spliced out and the exons get joined back together to form the mature messenger RNA that is then translated into protein, and this entire process is called splicing. Now, yes,
What happens to the introns?
Get
They just do nothing.
thrown away. Well, there's, yeah, you might ask, well, why would biology waste all that energy on just these pieces that we're gonna throw away anyhow, right? That's an excellent question. And, well, turns out that splicing doesn't always happen the same way. So what you're seeing here is, well, let's say you need to quickly silence a gene. You don't want a functioning messenger RNA. Well, you could shut down transcription, but then starting it back again is time consuming. So just make the transcript non-functional, so that's it, or function in a different way, because you could also make a change in splicing that will give you a different protein that works a little differently. So there's a lot of ways in which this is useful, and basically this is another layer of complexity that can give your genome a way of regulating how it works. And so let's say this intron, in some cases, okay, we don't, we're gonna make a different splice isoform, so you get something called intron retention. Now, in humans, the most common form of alternative splicing is called exon skipping, where you'll miss one of your exons. Intron retention, is extremely common in plants, and in humans for a long time, people thought it was just junk, but it's not really biologically relevant. So, yeah, we've had a little bit of uphill battle getting people to accept some of our research results and getting some of these projects funded as a result. So, why are we interested in alternative splicing? Well, it's a mechanism of regulation. It can give you different related protein. There's ramifications in disease, and that there are a lot of diseases that are the result of missplicing. And in fact, intron retention specifically is in cancer, there's a lot of intron retention going on, and for reasons that I'll talk about a little bit, but yeah, definitely there's a cancer connection there. So overall, it's increasing what your genes are capable of doing. And back to chromatin. So, because yeah, I was talking about chromatin, and now all of a sudden we're talking about mRNA. So what's the connection there? Well, turns out that as a gene is being transcribed, it's also being spliced at the same time. So what's happening at the DNA level is affecting what's happening at the RNA level. And so what we wanted to do is to uncover some of these biological signals that are present in the DNA and affect the process of splicing. And so a couple of years ago, we had a paper in genome biology where we just trained a regular old one-dimensional CNN, just nothing fancy, to distinguish between introns that are retained and introns that are not retained. And in our analysis of the network, we saw that there are a lot of transcription factors whose binding propensities are predictive of intron retention. And actually, we then looked at the literature, and a few of those had also a known association with the alternative splicing. Yes.
Yes, again, in this case, you're training your CNN in a supervised manner.
Yes.
Okay, you're not using foundation models.
Yeah, no,
Zero
this
shot.
was
Okay,
just
so it's just the CNNs in a
yes.
supervised manner.
yeah.
And what's your regular kind of split between training and testing towards, and is it cross-validated multiple times?
Well, what we did here was just an 80-20 split. You have to be when you set up your train test splits, you have to be a little careful in that you want to make sure that you don't have in your test set regions that are similar in sequence to the training set, because then there might be some subtle biases that creep in.
And you do it in a cross-validation manner? Multiple
No, we
times?
just had a
Wow.
fixed train test sprint. So, well, now we have DNA language models. So, well, let's try what those can do. So presumably the pre-trained language model is learning a representation that is useful, but what we're going to compare the DNA language model is to basically using this supervised chromatin state model as a starting point in the same way, where we'll chop the head off of this supervised model and fine-tune it to the problem that we're interested in. And so we took this same model that I just described and applied it, and what we're seeing here is a comparison of the fine-tuned say model compared to so that's the red. The blue is basically some other architecture that we trained from scratch, and then compare that to a model that is based on DNA BERT2. And so we're getting slightly better results with the model trained from scratch, and even better results with the fine-tuned chromatin state model. So we took the same model, we added a little bit of a convolution on top of it, although that wasn't strictly necessary, and then just a binary output. So logistic regression is all you need. It turns out that we can get almost the same level of accuracy instead of using the representation learned by the same model. We're actually using the actual output of the model. So remember, it's giving us a 20,000 dimensional output vector that tells you what all these transcription factors are doing, what the chromatin modifications are in that region. And then, well, okay, let's just use that 20,000 dimensional vector as input to our model, and we get something that works almost as well as the fancier fine-tuned version. And the advantage of that is that, well, you can look at the weights that the model has learned, and it tells you, well, which transcription factors it thinks are have a strong impact on this prediction problem. And so this is a list of the top 20 the top 20 weights that the model found. And what you may see, some of them are represented multiple times because, for example, yeah, this MYC transcription factor, yeah, they looked at multiple profiles were used in training the same model, so that's why a given transcription factor appears multiple times. And so actually for this, we didn't use the entire 20,000 dimensional output of say, we only used those outputs that specifically describe the transcription factors. And yet these are the top 20. And then, okay, we went back to the literature and checked, well, which ones had a known association with alternative splicing. So yeah, POL2 is not a transcription factor. It's just the RNA polymerase, but it, yeah, we kept it, but we kind of liked it there. So it was not surprising because a lot of transcription factors can bind the CPD domain of polymerase, and that's how it can interact with these spliceosomes. So that was, we thought, a good thing. Yes, Nasa?
The previous slide, I'm still thinking about that.
Yeah.
The slogan is logistic regression is all you need.
Yeah.
But actually, it's the foundation model that you also need.
Yes.
That gave you that useful representation.
Yeah.
Right.
Absolutely.
And then you use logistic regression as your final prediction layer.
Yeah. So logistic regression is all you need given a foundation model.
Right.
That same model. Yes.
So FC is
the fine-tuned version that uses this architecture. So POL2, AFF4, MYC, which actually has a connection to cancer. And in fact, yeah, I've been talking to Fayaz about maybe, but getting some cancer-specific transcriptome data that we can maybe get predictions that are more specific to intron retention in cancer. Because, well, cancer is a disease of the genome, but because of this relationship between chromatin and splicing, I think that creates a strong effect. So cancer, yeah, there's strong impact on chromatin, and that in turn will directly impact splicing. Yeah, CPCF, everybody's favorite protein, also has an impact on splicing. So yeah, a fair number of these top 20 already are known to be related to splicing. So that gave us good confidence that our model actually is picking up on biologically relevant candidates. So that's this first part. The second part that I'm just going to give a very rough overview is about a project we have that is currently ongoing in the lab, and that is to design our own supervised foundation model, but this time for plant genomes, because most of this work in deep learning and chromatin state prediction, the vast majority has been in mammalian systems. The only work I've seen in plants uses architectures from 10 years ago and rather small data sets. So for the longest time, in plants, there wasn't a data resource like ENCODE that would support the development of models like this, because putting together like something along the lines of what they used for, say, these basically 20,000 data sets analyzed in a consistent way, that's a lot of work. But even using this chip hub resource that was recently published, it still took us a few months to put together this data set so Arabidopsis thaliana and Rhiza sativa, that's Arabidopsis and rice, two of the primary model plant organisms. Now, Arabidopsis is kind of an interesting plant because it's a very compact genome. Yeah, humans, that's 3 billion nucleotides. Arabidopsis, just a tad over 100 million. So the intergenic regions are very short. Everything is nice and compact. Some other plant genomes, well, we're looking at corn. The corn genome is actually larger than the human genome, but there's tens of repetitive sequence there. So anyhow, we found this resource. For Arabidopsis, we able to get around 2,500 different data sets. Well, it's not 20,000, it's just 2,500, but that proved to be more than enough. So we trained, this one is a combo of a stack of convolutional layers, some transformer layers, and a prediction head, and again, predicting the same sort of outputs that Say did, so histone modifications, transcription factor binding, and DNA accessibility. And what we wanted to show is that this model learns a representation that is useful for predicting things related to gene regulation, like gene expression and the activity of enhancers. So I'll just jump straight, well, here. where we're using our deep plant model and two plant DNA language models, and we're trying to predict gene expression either by fine-tuning one of these plant language models or fine-tuning our deep plant. And so we're, again, trying to predict these over 3,000 expression profiles in Arabidopsis. And our deep plant model gives us overall better results than both of these. Although it's interesting, the distribution of correlations for AgRNT has this weird shape that we need to figure out what's happening there. So that's one nice result. Another nice result was for predicting enhancer activity, where again, this is just a simple binary classification problem. And again, we're getting better performance with our deep plant model than with the two DNA language models. And now, yes,
How does it compare to other non-plant-based generic DNA LLMs?
Compared to any of those, because at least in the publications that we saw, AgRNT and its evaluation on plant-related data sets was working better than
So they did a comparison with the generic.
yeah.
I see.
Yeah. Now, another good reason to pick our model is just how much easier and faster they were to train. So these are the training times of deep plant models compared to these DNA language models. And yeah, for the language models, we needed kind of A100s to do the work. Our model, just a more commodity GPU, worked just fine, and way. So what I was hoping to show you is that yeah, these supervised models are what we think they're a good platform for developing models for problems related to gene regulation. So that we have examples in both humans and plants. There was some interesting biology related to splicing, and so overall, I think that yeah, deep learning has its role in genomics, and as usual, it's always about what we can learn from these models that's relevant to biology. And with that, I'll thank all the students and colleagues involved in these projects. And this is Elron, who's the lab mascot. And with that, I'll take any questions.
Does anyone have any questions?
Yes.
So I see how for transcription factors, it's probably just finding a motif and finding it for the genome. For histone modification, you're doing basically the same to predict if there is a modification in a specific area, right? But I think histone modifications are like associated with diseases, so they might vary between people. Is that correct?
Yes. Yeah, and
So
in general, that is true for just about that could be true for transcription factor binding as well. Yes,
So when we predict the modification, it's more like an average, and it would not be taking into account that having a modification could lead to a disease. So
correct. It's assuming a specific reference.
Yeah.
Yes, and there's also other ways in which people are taking this to the next level, and that is because this is all bulk assays. So people have started applying this to single cell sequencing data to see how individual cells are functioning. So that is the next level of doing this. Although the problem there is that the data is incredibly sparse. So it always tends to be a little bit of an average.
because I think how I previously used something like this was I was working with GWAS, identifying single SNPs and then seeing if it is in a transcriptor factor binding site
Yeah,
or in a modification.
yeah.
So do you think it's still useful if we use this like
Yes, because the overall biology, the cell still functions overall similarly. So yeah, in disease, you have some aspects that are different, but the overall function is still the same. So I would agree that what these models are learning is kind of the broad strokes of how the genome is functioning. Yeah, part of what makes these models also interesting is that they have been shown to be useful at figuring out what the effect of mutations is, although I'm still not totally convinced by that, but
I have a more generic question.
Yeah.
When is a foundation model a foundation model? Foundation model. The results that you've shown seems to me are for very specific tasks, whereas the connotation of foundation model is that you can take a model and apply it to your own tasks.
yes, it's foundational within the context of gene regulation. So so far, these have shown to be, we've seen them, these models being useful for basically every gene regulation problem we tried so far.
Okay.
So I think
That's your definition of foundation model.
that that it learned something, learned the general structure of a phenomenon that makes it useful in a variety of tasks related to that domain. So yeah, it is not a general purpose foundation model in the sense of a human language foundation model. And we've seen some cases where the DNA language models work better when it was problems related to the nitty-gritty of how a specific sequence works. So yeah, it probably does. These don't have the general expressivity of the DNA language models, but in this more restricted context of gene regulation, which is still a pretty broad area, I think. Yeah, it's well, it's also the buzzword of the day
Yeah.
that gets people's attention, of course.
Yeah.
We
I'm
are
looking at a
guilty,
paper.
we are guilty of that.
I'm looking at a paper with the title, When is a foundation model a foundation model?
Uh-huh,
I'll share that with you.
sure.
And they also show similar kind of results that significantly smaller network gave them better results on specific tasks.
Mhm.
So they were questioning the
The need for
The-
these,
the hype about foundation models that
Yeah.
sometimes smaller models for
Mhm.
specific tasks, and a lot of people have shown that
yeah,
specific models for smaller tasks, smaller
Yeah,
models for specific tasks.
there's a lot of hype here and bigger is better and all that.
In fact, there was a recent paper just last week by NVIDIA which showed
Mhm.
small language models actually are better for specific agents. You know, the whole buzz about
Yeah.
agentic AI, that if you want to develop agents,
Yeah.
agents have very specific functions,
Mhm.
that small language models actually are better than large language models for agentic purposes.
Mhm.
Do you have a view on that?
Well, that makes sense.
Mm.
If you're probably better able to fine tune the model for that agent's purpose. It's steering a large model in a specific direction is probably harder than doing that to a smaller model. Yeah, thanks for that.
Do we have any further questions from the group at all? I suppose if you could, I suppose in terms of the first task that you presented, sorry, could you potentially reiterate for us again what the sort of route is towards helping patients with this sort of technology?
I haven't given thought to that. That was basically more on the basic science of the relationship between splicing and chromatin state. But yeah, if we were to maybe explore this in the context of cancer, that maybe there are therapeutics that you could apply at the chromatin level that would address splicing related issues that would impact the progression of the disease. That's just the pure speculation.
Sure. Thank you very much.
Hi, Professor Aisha. Really nice talk. Can I ask a question about that we are doing RNA sequence and non-long coding RNA sequence analysis, and we are looking into the alternative splicing, which one is just you mentioned. We want to see which part of which alternative splicing can regulate tumors development, and how do you think if we can apply the fine-tune the C model to increase to make the prediction more correct, more accurate?
Yeah, so for that, you would need RNA-seq from cancer samples, and what we did here was basically just a simple distinction between retained and non-retained introns. What we would do there is to have a more quantitative tissue-specific prediction problem that addresses the splicing in that tissue context.
Yeah, we do have the samples like related to the tumor and normal tissues, and we do have the the sequence from them.
Oh,
And
excellent. So in that case, yeah, just send me an email and maybe we should get in touch and
yeah,
we see how we can do something with that.
sure, will do. Yeah, thank you.
Okay.
The following question is like some kind of future work. So because you compare the DNA language model with your C model, right? So do you think you can do somehow use both two types of models advantages? I mean, combine the advantages of two different models to create some future work?
Absolutely. Yeah, in fact, yeah, what I kind of glossed over some of the results here that, yeah, in fact, well, if we're trying to predict what about 2,500 different outputs, and what I have here is a scatter plot of prediction performance across these different outputs, and what you're so this is comparing the DNA language model and the deep plant model, and what you're seeing is that yeah, it's all over the place. So that in some cases, actually, if you look at these green dots. This is open chromatin, and what you're seeing is that the DNA language model is doing a lot better than our model for that kind of experiment. So so I'm definitely interested in well, there's probably is a good way to combine the two models. I also want to understand why our model is not working so well for the open chromatin data sets. Yeah, what we tried and only somewhat worked is to just ditch the convolutional layers completely and just use a completely transformer-based approach, but so far we haven't gotten that to work as well as our current model that has a whole bunch of convolution layers followed by transformer layers. And yeah, we've been also playing around with some mamba layers, and so far that has also worked moderately well, but we've had some trouble with the instability of the training of the mamba layers, so we're still hoping to get that working, because I guess yeah, yeah, sometimes getting these models to work yeah, can be a bit of a challenge. I'm sure every one of you have their own stories about that. All right, excellent questions.
Fantastic. Well, yeah, thank you very much for a really interesting seminar. And I think with that, we'll call the seminar to a close then. And you're going to be around for a bit more time today, aren't you? In case anyone's got any questions or would like to.
Yeah, we don't have anything scheduled right now, and so if anyone wants to chat, I'm here.
Fantastic.
Okay.
Brilliant. Well, thank you very much, everyone.